Hi everyone, let's continue our discussion on the regime where mutations are rapid and which leads to the implication that there are more than two genotypes which coexist in an environment at the same given time. And this analysis is going to be slightly tricky as compared to mutations are regime precisely because of the reason that we have to take into account multiple coexistence of multiple genotypes in the environment at that particular instant. But before we try and work towards development of the expression corresponding to the speed of evolution in such an environment, let's try and intuitively understand via the representation with which we stopped the last lecture and understand what's happening in this population as time moves forward, starting from a population where each individual belongs to one genotype only. So let's think about this. We have at t equal to 0, we have all individuals at fitness f naught. This is the picture at t equal to 0. At some future time, a mutation event is going to take place and this population, one of the individuals is going to mutate and I will have an individual which is at f naught plus s. As soon as this mutation gets established, its numbers are going to increase and then selection acts and the numbers corresponding to this are going to decrease. But as the numbers of individuals which correspond to this particular genotype are increasing, one of the individuals belonging to this particular population is going to mutate and give rise to an individual which belongs to a genotype whose fitness is given by f naught plus 2s. And this keeps on progressing forward in time that each pool of individuals at the highest frequency, the leading edge as we call it of this distribution is going to keep giving rise to individuals of higher fitness. That is going to keep on happening. So, what mutation is doing here is that mutation is creating variability. Mutation keeps having these newer genotypes arise in the population and keeps widening this distribution that we have. On the other hand, selection is trying to narrow this distribution. Selection is trying to weed out the less fit individuals from among this distribution and that works towards removing individuals which correspond to this particular genotype, for instance in the picture that we have just drawn. And question that we should ask ourselves now is that suppose we started this particular experiment with all individuals corresponding to fitness f naught. Now, if we wait a long enough time, what does this distribution of individuals corresponding to particular fitness bars on the x axis look like when we have waited for a very long time? And one way to think about this is that mutations, when we have waited for a long time and observe this distribution, then individuals at the leading edge are going to be small in number by definition, and that is because these particular genotypes have only just arisen in the population. The mutation events that led to their coming into being have only just happened and they have not had time to flourish and spread through the population, even though they are the ones which have the highest fitness in the population at that particular instant. But because they have only just arrived in the population, their numbers are going to be smaller in number despite them being fittest in the population. Their numbers will rise with time, but at this time the numbers associated with those particular genotypes are going to be small. So, these numbers, the leading edge is going to be small in number. The lagging edge is also going to be small in number because these are the genotypes which correspond to the least fitness in the environment at that particular time. These are individuals which probably arose some time back. Once they must have been the fitter ones, but then and correspondingly their frequencies increased, but then fitness started acting, mutations kept on generating newer individuals which had higher fitness corresponding to them, and hence their numbers came down and now they are on the verge of being extinction. And something like this distribution will eventually meet that its numbers will keep on coming down, and while they are coming down, the numbers associated with these two and the subsequent fitnesses will be higher, and we can imagine that the lagging end individuals are also going to be small in number. So, if we think about this in those terms intuitively, what it tells us about the steady state picture of the distribution, if I take a snapshot of the population at any time t, the distribution of individuals across the different frequency fitness bins would look something like this. Would perhaps look something like this. X axis again represents fitness and y axis represents number of cells which correspond to a particular fitness. And what's happened is that these individuals, they are the fittest individuals in the population. So, they are the fittest, but still their numbers are small because they have only been in the population for a very short time. At the time that this snapshot was taken, it has not been very long since these mutations have come into existence in this environment. Hence, their numbers are small. Before these individuals came into being, these were the fittest individuals. So, before these two came into being, these were the fitter genotypes, and hence their numbers, their frequencies were increasing in the population. So, and hence they are represented by the highest frequencies among all distribution, among the distribution of the fitness among individuals. And at the lagging end, these are on the verge of extinction. And if you compare these two graphs, you can imagine that after we wait a while, this plot would start to look something like this, where the numbers associated with this particular genotype will decrease such that the number corresponding to this genotype is represented by this small bar here. So, at steady state, it sort of makes intuitive sense to anticipate a distribution such as this. And this distribution, however, is not steady in nature. This is not steady in nature because selection is continuously acting to remove these genotypes from the population. Selection is also acting to increase the frequency of these genotypes in the population, whereas mutation is pushing this wave forward. So, at steady state, this distribution acquires a moving wave kind of a phenomena, which is perhaps best represented by this graph. So, at t equal to t1, the distribution looks such as this. But then we have new mutations arising from this particular genotype and everybody is moving to the right. At the same time, selection is acting to ensure that these genotypes are eliminated from the population, and selection is also acting to make sure that these genotypes flourish in the population and the frequencies associated with them increase with time. If that is the case, then if I move forward in time to t equal to t2, what would have happened? Because selection is acting to increase the frequencies of these genotypes, their numbers would have gone up in time. So, now in this snapshot, this particular genotype was the mean genotype which was at the highest frequency. That has changed now because selection is acting to reduce its fitness, selection is acting to increase the frequency associated with this genotype, and its frequency has gone up, and now the genotype which is maximally represented in the population is now this particular genotype. At the same time, the frequency associated with these two genotypes has also increased as we move forward in time from T1 to T2. But what has also happened is that the leading edge at T equal to T1 has produced a mutant which is carrying a beneficial mutation. Hence, the leading edge is now this bin where there were no individuals at t equal to t1. That has happened because of this leading edge, the earlier leading edge producing a beneficial mutation. At the same time, the lagging end has been acted upon by selection, and that is no longer present at t equal to t2. So, what this again tells us is that the steady state distribution of individuals in this population can be represented by this moving wave kind of a phenomena, where this shape, that this population has acquired this shape, and this shape is just moving to the right with time, leading to increase in fitness. Again, the assumption that we have made here is that beneficial mutations are always available to the cell, they don't dry up. So the mean fitness associated with the population keeps on increasing as we move forward in time. But something that we did a couple of lectures earlier was that in the initial phase of evolution, we saw that fitness increased linearly with time and then saturated. So if you are only talking about that linear phase of evolution, this result holds, that beneficial mutations do not dry up and it keeps increasing. The mean fitness keeps increasing with time in a linear fashion. So if this wave-like property of distribution of different genotypes in the environment makes intuitive sense to you, what we'll do next is try and derive an expression for speed of evolution in a setting like this. And it should be intuitive to note that speed of evolution, the rate at which the mean fitness of the environment is changing with time, is actually just equal to the speed with which the wave is moving that we have just drawn. So essentially what we are interested in is that how fast does this wave move. Okay? So that's something that we'll work towards now. So again, we are interested in the regime of rapid mutations, and we want an expression for speed of evolution which is also equal to speed of the wave that we just described. Okay. One thing to note is that since this distribution is moving at a constant speed, that's our hypothesis, if this is the distribution at t equal to t1 and then at t equal to t1 plus tau, what has happened is that every bar has gotten shifted to the right exactly. So if this is the mean fitness f0, this is s, this is f0 plus 2s, 3s and 4s. Now f0 has shifted to f0 plus s, that's the maximally represented bar now, and then you have plus 2s, 3s, 4s and 5s, and subsequently this is minus s, this is minus 2s and this is minus 3s. Now you have f0, f0 minus s, f0 minus 2s. And f0 minus, what has happened between these two time events is that f0 minus 3s has been acted upon by selection, which has led to its numbers corresponding to this particular genotype being reduced to 0, and mutation events in this particular population f0 plus 4s have led to individuals belonging to a fitness of f0 plus 5s getting established in the population. So one way to notice that in this tau time, the wave has moved to the right exactly by s units. We can think about this in simpler terms, that every individual in this population, it's almost equivalent to think that every individual in the population has jumped s units in fitness in this time tau. So speed of evolution in a setting like this is just equal to s divided by tau. Because the mean fitness has increased by s, and we can say that because fitness of every individual in the population has increased by s, hence the mean increase is also s, and that change has happened in time tau, which gives me the speed of evolution, which is the rate of change of mean fitness with time equal to s by tau. And what we will do now is try and develop an expression for this tau. We do not know what this tau is, and that is what we will try and develop and quantify next. To do that, we first understand, we first realize that we do not know the distribution, we do not know the exact distribution of the genotypes at any particular time in t. Even though we are drawing frequencies, we do not exactly know the numbers associated with each fitness. We have an intuitive hypothetical understanding of the distribution, but not in a quantitative sense. So, what we will start with is a few assumptions. We will assume a steady state distribution of population at any time t, and in that distribution, let me just draw this. We are not sure how wide or how narrow this distribution is. We do not exactly know anything about this distribution, except for the fact that intuitively we feel that this should be the structure of the population at any time t. This is fitness, number of cells. If this is the case, and this, so we will assume a symmetric structure of the population. If we assume a symmetric structure of the population, that automatically implies that the middle histogram here, which is the highest in frequency, represents the mean fitness. Because this is so, because we are saying that the structure is symmetric about the mean, then this is f naught plus s, this is f naught minus s, and we are saying that the number of individuals belonging to each are equal in number, and hence we have the same number of individuals which are at f naught plus s and the same number of individuals at f naught minus s. So, if we add these two together, the mean fitness will be exactly cancelled out by each other, and the mean fitness will come out to be exactly f naught. Similarly, this bunch of individuals has a fitness f naught plus 2s, and this bunch of individuals has a fitness f naught minus 2s. But because these two numbers are equal, because of the symmetric rule that we have imposed here, that means that the mean fitness of these two bars is f naught exactly. The plus 2s and minus 2s cancel each other exactly, and we get mean fitness equal to f naught. So, we take these bars in pair, and what that will automatically lead us to is the fact that the mean fitness of the entire structure is just f naught. All right. Then we assume that the leading edge is q mutations away from the mean. What that means is that this particular genotype, which is the leading edge, is from the mean at this particular time q mutations away. That means in fitness terms, each of these q mutations confers a fitness change of magnitude s. So, the fitness change between the fittest individual at the leading edge compared to the mean fitness is equal to q times s. Okay. It is important to know that this q is something that is yet unknown. I do not know if q is 4, 5, 10 to q could be anything, but at this point we do not have an idea of how much is q. And since we are interested in finding the tau, because we need tau to be able to comment on the speed of evolution, we will find that tau and q are quantities which are pretty closely connected to each other. All right. So, with these assumptions, now let us try and develop this further. How do we think about this? We think about this in the following sense. So, let me just redraw the graph again. and the less fit individuals, this is F naught. First, I am going to focus on the leading edge, and let me call the time tau. We have already defined tau. Time tau is the time in which this wave moves exactly S units in this fitness domain. So, from here to here, right now the distribution is from here to here. This is F naught plus S, 2S, 3S, 4S. So, it moves from F naught minus 4S to F naught 4. This is the range of the wave at this point. At time t, this wave moves exactly by S units on this axis. So, at time t plus tau, the wave will have a structure which is distributed across these fitnesses, and hence we say that the fitness of every individual, it is almost as if the fitness of every individual has increased by S, and so the speed of evolution is given by S by tau. But can we comment on this tau? What we are going to do is comment on the value of this tau in a couple of different ways. The first way that we will comment on tau is that how much time does it take for the leading edge to no longer be the leading edge. Which means at the time that this snapshot was taken, this particular genotype represents the leading edge of the wave. But eventually we know that another mutation is going to happen and a new genotype will arise, and that genotype, when established, will form the leading edge of this wave. And when that happens, this particular genotype, which is currently the leading wave, will no longer be the leading wave. We know that that time when the leading edge is defined by this particular genotype is exactly tau from when this picture was taken. So, the first thing we are going to try and do is estimate that how much time does it take for a beneficial mutation from this genotype to generate this particular genotype of a higher fitness, and then this mutation to get established. That is the first thing that we are going to start with. So, how do we think about this? Now, because the leading edge is the fittest individual in the population, it is reasonable to expect that its numbers are going to grow exponentially, because at that instant there is nobody more fitter than that particular individual in the population. So, for the leading edge, we can write that NT is equal to N naught e to the power ST. This is a general formula that holds for any exponential growth. For the particular case that we are interested in, we can write for the leading edge, we can write that NT, this is the number of individuals belonging to leading edge, is equal to N naught. Now, what is N naught? N naught is the number of individuals of the leading edge at the start, which is equal to 1 upon QS. Because remember, the advantage that the leading edge holds, over the mean is exactly equal to QS. If the advantage that the leading edge holds over the mean is equal to QS, that means to become established, it needs QS 1 upon QS number of individuals in the population. So, when the leading edge has 1 upon QS number of individuals present in it, that is when it becomes established. After that, its growth is exponential, and again the growth factor is just QS into T. That is the rate at which the leading edge is growing. What is the rate at which it is producing mutations? So leading edges numbers varies like this. What is the rate at which leading edge is producing mutants, and that's just equal to NT times U. That is the rate at which mutations are happening at the leading edge. And next is, among all these mutations that are happening at the leading edge, how many mutations are actually getting fixed or established? So among all leading edge mutations, how many get established? And that is just equal to NT times U. This is the total number of mutations that are happening at the leading edge. We know that the probability that these mutations will get established is just equal to Q times S, right? Because the probability that a mutation which has an advantage S over the mean is equal, the probability that the mutation carrying an advantage S over the mean survives is equal to S. So the probability that this mutation which holds an advantage QS over the mean survives is also equal to QS. Now what we want here is that, so this is the total number of mutations which are getting established. We want, what we are interested in is that we want the time it takes T for the total number of mutations from the leading edge which get established equal to 1. We are interested in solving this equation for the time such that the total number of mutations that get established is equal to 1. And that is so because if the total number of mutations which have established from the leading edge is equal to 1, what that means is that there is a beneficial mutation which happened from the leading edge and that mutation has gotten established. What that means is that the leading edge is no longer the leading edge and has now been relegated to the second bar on the distribution that we have here. And that time it takes for the leading edge to produce a mutation which gets established is also equal to tau. This time is going to give me a value of tau. Okay. So this is slightly involved and we'll continue this discussion in our next lecture and arrive at a value of tau in a couple of different ways, and then eventually we'll solve for tau, which will give us the speed of evolution for a case like this. We'll pick this up in the next lecture onwards. Thank you.